Hello, is it working? Okay. So first of all, they're telling me something in the back, no? Okay, thank you all for coming here to listen to our talk. So first of all, I'm going to introduce my friend, William Blevins. He's a PhD student in genomics in my former lab in the PRBB. And he's also a very avid maker, and he enjoys tinkering with all kind of different things.
I'll introduce my compañero as well, Jose Luis. He has finished his doctorate in genomics, and he's worked with all sorts of organisms, including fat-tailed dwarf lemurs and the Iberian lynx. And he's an avid historical fencer. They use the classic real style like it used to be, and archery as well. It's one of his big hobbies.
So we're going to start with a little video.
Okay, so there's no sound apparently, so I'll do the sound.
What do they got in there, King Kong?
So, okay, I don't know where were you when this movie was released, but I was 9 years old and I wasn't allowed to go to the cinema. So I went, because the movie was for 13 years old, so I went with my parents. For me, it was quite an experience. It was one of the big reasons why I got interested into science, and obviously there were dinosaurs in it, so the mixture couldn't fail. Before going any further, we have to say that we want to make this presentation interactive. So throughout the presentation, we're going to ask you questions, and you can answer them with your mobile phone, and we will see the answer in the screen. Okay, so if you please would go to this website, menti.com, and you'll have to introduce a code, a 6-digit code that will appear on top of some slides. So when you see a code, please go there, insert the code, and you can answer the question, okay?
Right, so before we get started, we'd also like to give you guys a disclaimer. We're not the kind of scientists that make rabbits with human ears coming out of their backs, or monkeys with 7 legs, nor do we really kill animals in our research. We're computational biologists, so we spend most of the time just looking at a screen. We're code monkeys. We're also going to bring up some controversial topics and kind of play with them, play devil's advocate. These views don't necessarily reflect our own, so please don't get mad at us. If you want to talk with us after, we'd be glad to discuss all sorts of different topics, but don't interpret this as our views, please. Also, dinosaurs are awesome.
So we're going to start a bit with Jurassic Park. As you all know, the novel was published in 1990 by Michael Crichton. So he's one of the fathers of the so-called techno-thrillers. So he gives an immense amount of detail to all his novels. He gathers a lot of information, and he gives you very precise information, and this allows you to get into the novel and gives you a lot of credibility. So the environment where this novel is set is the US in the late 80s, early 90s. So what was going on in the States is that there were a lot of biotech companies that were starting or going very strong. And you can imagine that biotech companies go all the way through pharmaceutical companies to, biotech. Even artificial snow is considered a biotech company. So also drug companies, farm crops, etc. So Michael Crichton was inspired by the research of this guy here. Maybe you know him, maybe you don't. He's Jack Horner. He's a very well-known paleontologist. And he was deeply involved in the saga. So Crichton knew when he wrote the novel, that dinosaurs were warm-blooded, and this is reflected in the novels and in the movies. He also knew that raptors had feathers. I'm sure you all know that right now. And what is interesting to me is that he also advised the movies, and actually it was Steven Spielberg who convinced this guy to not include some of the research, because who would have been scared by a raptor with feathers, right? So also I want to mention that sometimes this inspiration goes in both directions, because just after the release of the movie, Jack Horner got an insane amount of solicitudes, like
applications
for doing their PhD in their lab. So it was an inspiration to many, many people. Right. But today we're not talking about paleontology, we're talking about genomics. And I want to introduce very, very few concepts so you can follow throughout the presentation. So first of all, what is a genome? We have here Mr. DNA, which will explain it to us. So basically, in all of our nucleated cells, we carry a pack of information that contains all the necessary information to build an organism from scratch. It contains the codification to create proteins, that all the small units that perform all of the biological functions. And this information is encoded in the form of 4 different nucleotides, the A, T, C, G. You've seen this a million times, but this is just a small reminder. So, for example, in humans, we have 3 billion of these letters packed in our code. So it's not a trivial problem to work with in genomics. So we work with very large amounts of data. So here goes our first question. So
get out your phones.
You can get your phone now. The code is 875831. So which organism do you think has the largest genome? Can we see this here? Yeah. Do you think is the rice? Human, we have Neil deGrasse here. Yeast.
All right.
Amoeba,
We'll
rice.
give you guys another 20 seconds.
Human. You can vote from home too. I mean, this works.
For people watching streaming?
Yeah, watching streaming, I know.
Okay. I think we have a clear winner.
Yeah, I think we have a lot of biologists in the audience. Or they're cheating.
I don't think anybody can use Wikipedia that fast.
Okay, so you can keep voting, but we'll cut it here. So, what?
Go back to the public.
Yes. So yes, the largest genome is actually the amoeba. Most people is surprised by this fact. I know that you in the audience are not, because most of you got it right. So perhaps because we're human, because we're men, always think we have the largest something, but this is not the case. And actually, the human genome is kind of average in size, at least. You can see it on the top. You have the distribution for each group of animals. So the size of the genome doesn't really tell you about the complexity of life. So, for example, an amoeba that some people can conceive it that not very complex is actually one of the largest genomes. Okay. So very short and incomplete story of genomics to get you on track. In 1953, Watson and Crick unraveled the 3D structure of DNA, and they got a Nobel for that. And sadly, Rosalind Franklin was also involved in this work. Not sadly he was involved. Sadly he didn't got the Nobel because she died just before getting it. So that was a pity. And the first sequencing was done by this guy who also got the Nobel. He actually got 2. He's one of the few people with 2 Nobel Prizes. He sequenced the first virus in 1977. This little thing there with nearly 5,000 base pairs. And Jurassic Park is set just before we were able to sequence a bacteria, which is around 1,800,000 base pairs in 1995. And interestingly, in 1990, it started a very important project for science and for humankind, really, was the sequencing of our own genome, the human genome, that started in 1990. And it took 13 years to just complete the sequence, okay? And it cost almost $3 billion. So imagine the scale of how hard it is to work on these things. Okay, so because we're huge nerds, we looked through the book, and this is one of the things I love about Michael Crichton. He gives a lot of detail, and he gave actually the model of the sequencers they were using at that time, and they are real sequencers. And we got to the manual of the machine. So we look at what was the output of this machine. It was capable of sequencing 800 base pairs per hour. Okay, and they bought 24 machines in the ENG company, which, as you all know, is the fictional company in Jurassic Park. And we made very simple math, and we calculated that based on the expected size of a dinosaur genome, it would have taken 15 years to sequence just one of the dinosaurs, and not in a very good quality. Why I'm telling you this? We're not talking about what is wrong, what is right. It's just a comparison to make you understand what is the technology we have today. So this is one of our latest machines. It's an Illumina HiSec 10. This is just one machine, and you can see that we are sequencing orders of magnitude higher than in the 80s. So 2, 5, a lot of zeros, base pairs per hour. This equals 18,000 human genomes just in 1 year. So imagine the breakthrough we have had. Okay, this is more or less what you get when you assemble a genome. The correct order of all your nucleotides, A, C, T, G, boring. This doesn't really tell you anything. You have to, for example, look for where are the genes. I highlighted here the MC1R gene. First, you have to know where the genes are. You have to understand what are their function. You have to see is it found on other organisms. So this is a huge work. So remember this name, the MC1R gene, because it will play a part in the story later on. And here, we go back to another question. So how feasible would it be to resurrect each of these extinct species? So you should have a bar, no? And you can move the bar for each one of the species.
So 1 would be not at all feasible, and 10 would be totally feasible. What do you think?
Wow.
Only one person has voted so far, so...
But he's very optimistic.
Yeah. We'll give you guys a little bit of time.
Can we put it bigger? Or? Yeah.
There we go. Okay, we got 8 participants. We'll give you guys a little bit more time. But we can already see some trends happening here. Looks like you guys don't think T-Rexes are that easy to bring back.
You don't like the T-Rex or what?
All right. That's interesting.
Okay, I'm happy because the audience is very, very well informed, I think. I like that. So, okay, what was the result? Well, more or less. So the result was, okay, you think the T-Rex, we cannot bring him back, so, okay, sadly, yes. We have a lot of sequencing technology, we have a lot of output, but sadly, we cannot bring back dinosaurs, sorry. So why is that? As you probably know, or you probably don't know, the limitation here is the survival of the DNA, okay? DNA is a very, let's say, fragile molecule, and it deintegrates with time. And actually, this experiment that is shown in the movie was tested in real life a lot of time, and there are a lot of publications about this. And sadly, they were never able to recover insect DNA or obviously dinosaur DNA, even with very recent samples from 20 or 25 years. So apparently the amber is not a very good material because it's very permeable to water and stuff. Does that mean we cannot bring back anything from the past? Luckily, no. We can. So there's been a lot of ancient DNA projects, that's what we call them these days. For example, we were able to sequence Ozzy, also called the Iceman, and we gathered a lot of information about that historical period. Also, we've been able to sequence mammoths, Neanderthals, and the record right now is a horse found in the permafrost, which was 700,000 years old. Okay, obviously the DNA is degraded. We cannot recover the full sequence, but we can gather pieces and work with that. Okay. So really, Jurassic Park is not about whether we can or we cannot bring back dinosaurs. It poses a lot of very interesting questions in what I call biology of resurrection. We can come up with another term, that doesn't matter. So, for example, are the creatures we bring back actually dinosaurs, mammoths, or Neanderthals? And this is treated in the novel in the form of this character, Henry Wu, and he's always telling Hammond or Masrani in Jurassic World that the creatures we are bringing back are not actually these creatures because they are lacking a lot of things. So DNA is not everything in biology. And for example, what do you think that these creatures are missing? So now we know that the microbiome, for example, is all the bacterial community is also very important. We cannot bring back that from the past. Also the social structure and the culture of the different species, that is very important. And well, I'm just going to say this because we work on this, but also lineage-specific genes, so genes that are only found in one or very few species. So if you, in the movies and in the novels, they use other species to complete the genome, but there is a fraction, and we think it's a very important fraction, that is only present in one or few species, and these are actually what makes a species, some of the specific traits of every clade. Also, why would we bring them back? I mean, we need a very good reason because this is not...
They're awesome, that's why.
Well, that's also, they're awesome, it's a good reason, maybe for us, but if you go to the government, hey, I want to bring back a mammoth just because it's awesome.
Okay.
So I think we can learn a lot of these creatures. For example, from mammoths, we can learn about adaptation to cold. We can learn why some species went extinct in the first place. We can learn how to improve our conservation policies, etc., etc., etc. Should we do it? How much would it cost? So if you're into these kind of questions, I recommend you this book from Beth Shapiro. She's an ancient DNA scientist working in the States, and I can recommend you this book. In science fiction, these questions have also been addressed, for example, in this trilogy, the Neanderthal parallax, where there is a wall with Neanderthals and another one with humans. In each one of the walls, only one of the species survives, and their societies are completely different, but what is more important, their mind structure is also different. So this is one of the reasons why we would bring back a Neanderthal community, not just one individual, if it was possible. To learn more about what makes us humans and to have another view from another humanity, because in the end, they still belong to the Homo genus, so they are also humans, and we can learn another way of thinking that we haven't really seen. So this is how we conceive Neanderthals today, thanks to a lot of work from archaeology, a lot of work from sociology as well, and also from ancient DNA. If you've seen representation of Neanderthals at the beginning of the century, they were like beastie creatures. They were almost ape-like, and this has been changing throughout the time. We have been giving them feelings, social organization, language, etc., etc. And with the genome, we learn quite a few things. For example, you remember this gene, the MC1R, is actually one of the genes responsible for hair color. And we saw that it carried a mutation present in Neanderthals that provided them with red hair. So we know now that some Neanderthals, were red-headed. And this is why they are depicted here like this. And perhaps the most important thing we learn about Neanderthals sequencing their genomes is that we breathed with them. We had sex, and not just casual sex, because that doesn't leave a mark on the genome, so we breathed with them for quite a while. And I'm going to ask you, if you take this guy and you dress him and you find it in the metro, wouldn't it look like Tom Hardy? I mean, I can tell the difference.
Yeah. I mean, it's very close. I mean, looking out in the audience today, I think I see a couple of people with Neanderthal DNA as well. But that's okay. I have Neanderthal DNA in me, and I think all of you do as well. Actually, I know that I have at least 268 Neanderthal variants in my genome. So a variant is if I look at a very small region of my genome, I share something in common with Neanderthals. And I took this genotyping assay from a company called 23andMe. I'm not affiliated with them. They're not sponsoring us. So if you want to do this, I won't get any money for it. But you spit in a tube, ship it off, and they do a a glance at your genome. They look over I think, 500,000 individual letters in your genome they're looking at. So it's not a full sequencing of your genome. And they counted 268 variants, and you can see on my different chromosomes where those variants lie that I had in common with some of the Neanderthal DNA that we've sequenced. And they put in this nice little graphic in the bottom, 2.4%. Well, mammoth? No, I'm not 2.4% mammoth. I don't know why they use that image. But I'm also not exactly 2.4% Neanderthal. They say this on their website. We cannot tell you exactly the precise amount of your genome that comes from Neanderthals. So they're doing this mostly for entertainment purposes. It's educational. You learn a bit about yourself and your ancestry. But everything you learn, you have to take with a grain of salt because they're not giving you your full genomic sequence. So how does this work? Well, first you get a kit for $99, or I think now it's almost $200 or euros or whatever. And you spit in a tube. You have to spit a lot. You have to fill up the whole tube. And then you put it in a box and you ship it off to a lab somewhere in California where they take out the tube, they extract the DNA from your saliva, and they copy it a bunch of times, and they put it onto this chip that has basically a library of all these different variants that they want to look at in your genome. And if you have that variant, that little spot on the chip lights up, and they know that you have that variant. So it's great to know that I'm not 2.4% Neanderthal, but what else can I learn from this? I paid $99. I want to learn something about myself. Well, I found out that they think I have a 61% chance of being able to detect asparagus odor in urine. They got that right, I can. And that I have a 29% chance of not having a lot of 29% chance of having a lot of upper back hair. I don't. You guys can check afterwards if you want. I don't have a unibrow, but I do have to take out some hair sometimes. My earwax type is pretty dry. Eye color is light. But you can see the predictions in some cases, like bitter taste, 51% chance to 49%. They don't really have a good picture of what's going on just looking at my genome. So there are some mysteries still. Just sequencing somebody's genome doesn't tell you everything about them yet. What else could I find? There's more. Well, they give you a summary of your ancestry. And since I'm from the United States, which is the self-proclaimed melting pot of the world, I expected this map to be lit up, and it is. But I'm still 97, according to their prediction, 97% of European ancestry. Some people get this back, and it's completely different to what they expected, which usually means somebody got cheating in their family tree.
Yeah, Granny got wild.
Granny got wild. But I do have some interesting things, some Native American, Middle Eastern, Northern African, and Ashkenazi Jew, which confirms something that my grandparents told me, that we had some Sephardi Jew in our lineage, which might be good for planning to apply for Spanish nationality with the recently approved program to... Anyways, that's a different story. Yeah. They can also tell you things about your health. The FDA had put a ban on this kind of information for a while until they were able to sufficiently show that people wouldn't freak out when they received this news. But they can test for potentially dangerous variants. And now the US government is allowing them to disclose this information to people who pay for this service if they want to see them. So they ask you, are you sure? Are you sure you want to know? And I said, yeah, tell me. So I found out that I don't have maple syrup urine disease type 1B, which is kind of disappointing because that sounds interesting. I don't have Tay-Sachs, I don't have sickle cell anemia, or at least they didn't detect the variant that's associated with it. So again, everything with a grain of salt. So yeah, I was pretty happy. I got the test results back. All right, I'm good. And they can also test if you're a carrier. So even if you won't actually suffer any symptoms, they can test to see if you are potentially could pass on this disease to your kids. So we're going to do another quiz. And I'm asking you guys, so it's a different code. You'll notice it's 787752. And we're asking, would you like to have your genome sequenced? So we got a couple of people answering already, but you can't see it yet because I did something wrong. Okay.
Ooh, bold. No?
All right. So now we can see. Got 86% of you saying maybe in the future. Okay, we got, 6% saying you already have. That's
yeah,
nice.
we have one working in Illumina there. Yes, I already have.
All right. So a lot of you guys are optimistic about having your genome sequenced in the future. I'm glad you guys are curious. Okay. Well, science fiction is chock full of examples of dystopian futures where sequencing is pervasive, where the entire population has been sequenced and the government has everybody's genomes on record in a catalog somewhere. Gattaca is one of my favorite examples. And in this future, parents are given the choice to select embryos. So they go into a fertilizing clinic and they're able to screen out all the embryos which have potentially dangerous variants, so diseases or disposition to addiction or all sorts of undesirable traits. In the case of the protagonist Vincent, his parents decided to conceive him naturally. They threw the die, and once he was sequenced, they found out that he had some negative variants, and so he was basically discriminated against because of his genome. So the interesting implications in this particular work were that just having our genomes on record somewhere would inherently lead to discrimination. We already discriminate in so many ways. Do we really need another way to discriminate against each other? And there were also some interesting, you want to comment on the...
Well, this is just a curiosity. Maybe Genome Eugene Morrow is suggesting these words to you, Genome Eugene tomorrow, like eugenics is coming. But we also found another interesting thing. This is for the more geeks in the audience, if you want to try it at home. If you take Gattaca and you write it 3 times and you try to translate it as if it was a protein, you'll see some interesting things. That's homework.
Yeah, homework for tomorrow. So one of the things that we're going to contrast are selection versus modification. In Gattaca, they were merely selecting for the embryos which did not have these deleterious variants. Whereas in modification, you're actually going in and changing things in the genome. So the analogy that we came up with was with selection, you're taking a bag full of dice and you're throwing them on the table, but then you're selecting all the ones that have 6s. Whereas modification, you throw all the die on the table and then you go on with a permanent marker and you color in all the dots that you want. The end result is essentially the same. Another awesome science fiction work by Aldous Huxley is Brave New World. Way ahead of its time. They hadn't even discovered the structure of DNA yet, but already they were talking about nature and nurture. So not only were they putting in mutagenic chemicals to modify the genomes of fetuses and embryos, they were influencing their future behaviors by conditioning them with audiovisual stimulus, by giving them electric shocks, so basically controlling the nurture part of the equation. And in this dystopian future, nobody's born naturally. Everybody comes out of a factory, and society is divided into castes of alpha, beta, but it goes down the list. You don't want to be an epsilon, you want to be an alpha. And it's a lot more dystopian than Gattaca, I guess. And there are so many amazing, we can't fit them all in here, dystopian novels about genetics. They all have interesting perspectives and very poignant criticisms of genetics. And they actually help us, both as scientists and just as society, to avoid these dystopian futures. There's a talk coming up, I think, at 4 o'clock about does science fiction help us avoid dystopian futures? And I'm going to attend that because I think it does. So let's go on to the next question. What words come to mind when you see the term genetic engineering? So in this question, you have to actually type in words. And I think you get 3 words each. So what words come to mind when you see the term genetic engineering? Let's see what you guys say. And we don't have a filter here, so please keep it PG. Clones, futures, all right.
Clones.
I'll give you guys a little bit more time since you have to type something in.
Happening, I like happening. Freak show.
Dystopia.
Mad scientist.
All right. Real superpowers. Ooh, yeah. Transgenic. Health. Elimination. Cloning. Future.
Snips.
Oh, do we get SNPs?
Yes.
Nice. All right. So, yeah, we haven't talked about clone control yet, but...
Pet dinosaur. Okay, Will, you're not allowed to answer.
My hands are off the keyboard. And eugenics, yeah. All right, cool. I'll give this another 2 seconds. If you guys want to take a picture real quick, you can. Or I'm sure it'll be on YouTube. So it looks like clones and eugenics and superpowers are in your guys' minds. I like where your heads are.
CPR.
Okay. Well, why would we want to modify the DNA of an organism? I think some of you guys are most interested in the last point here, from superpowers to clones. That's great, but it can also be used for more practical things like curing genetic diseases, halting the spread of AIDS, killing cancer cells, improving the yield of food crops, slowing or stopping the aging process, or like you guys would love, creating an army of half spider, half octopus creatures to rule the world. That one's probably going to come after the others. So should we modify genomes? I'm going to sidestep that and just say, well, we already have been modifying genomes for millennia. We've been modifying genomes using very blunt instruments, very slow instruments like selective breeding. You can see so many food crops used to be wild varieties of plants that had very little nutrition, huge seeds. They were just not easy to eat or cultivate. And we've bred them over hundreds and thousands of generations to have bigger fruits, bigger grains. And that's a natural process, I think most of us would agree. We've also, in the 1960s, used radiation to speed up this natural process. So we used atomic radiation to introduce mutations at a faster rate into food crops like the ruby red grapefruit. There's a disease-resistant cocoa plant. And these were created using selective breeding, but also using a little bit of atomic radiation to throw the dice a little bit faster. In the 1970s, we started using some transgenic technology where we were able to take genes from one species and give it to another species, maybe giving it some sort of disease resistance or cold resistance. But now we have a new tool in our shop, and it's called CRISPR. I don't know if any of you guys have heard or seen CRISPR in the news. If you haven't, you will. It's all over the place in the scientific community, and it's breaking into pop culture as well. Luke Cage recently featured a little snippet of CRISPR in its storyline. And let me tell you what CRISPR is. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. That's why nobody says that, they just say CRISPR. And it was discovered in, I think, the late 80s. As part of a bacterial immune system defense. And what it does is it protects these bacteria against bacteriophages, these viruses that infect bacteria. And it allows the bacteria to detect when viral DNA gets into their cells. They have a library that they can check, and they say, all right, I recognize this virus. I'm going to cut it into little pieces, and that way it doesn't hurt me. And it's a lot cheaper and faster than any of the previous methods that we've been using. Even some more recent things like zinc finger nucleases, which were a lot harder to develop and took a long time and were very expensive. And if you want to learn more, This website has so much information. So what can we use CRISPR for? Well, one example of the many things is we can fix dangerous mutations without changing the DNA of future generations. So you have an adult who has some sort of genetic disease, and we can send Cas9, which is an endonuclease it works in conjunction with CRISPR. Basically, we send it in there. It's an elite squad of these little proteins that go in, snip out the bad DNA, fix it, and you're fine, you're cured. I think everybody wants that, right? Just you get one injection or a couple injections and you're cured of this disease that's been haunting you your whole life. In the past, less than the past decade, half a decade, funding associated with projects that use CRISPR has skyrocketed. And this graphic only shows you up to 2014. In the last 2 years, it's been exploding. And this is going to lead to all sorts of innovations and discoveries as we're constantly improving this tool. We discovered it in bacteria and it does its job there, but when we repurpose it, we can make little tweaks to it to make it even better. So what happens now? Well, scientists are going to continue to propose new experiments, and they have to put these experiments forward through ethics committees, and the ethics committees weigh the pros and cons and either approve the project or say, no, that's not going to happen. We're going to have new discoveries, new technologies, and probably new laws and regulations that'll restrict the use of these things. Although it does have to be said that the genie is out of the bottle. Now that we know how CRISPR works and it's so cheap and easy to use, you can actually go out and buy a CRISPR kit online. If you want to do some genetic engineering and manipulation at home, you don't really need that much. You spend around 200 euros and you can make something completely new at home. I don't think you should. But just prohibiting it legally might not be so easy now. And hundreds of experiments using CRISPR are already happening all around the globe. There are at least 4 labs working with CRISPR and human embryos. And there are probably going to be at least a dozen before February.
Okay, so we want to end with a positive message. So, as you know, the motto of ENG is we make your future. And we want to argue that with all these breakthroughs in technology, in genomics, in CRISPR, etc., we think the future might be happening now. The future is now. And you, as a scientific, as a science fiction readers, you have a head start from the rest of the society in these matters, because you know, you've read a lot of dystopia, so you know what could happen, what could not happen. And we as a society are going to have to make a lot of decisions about this data I was going to say play, but not playing, we're working with. So it's important to take informed decisions. And one of the ways of spreading this interest in these topics is actually through science fiction. And I think we're going to leave it here. Thank you for listening. Hello.
My name is Ileana, I'm from Romania, and my question to you is, up to what point can we do these modifications into genetics without transforming ourselves in something else? I mean, still being human.
That's a really good question. So if you're asking about speciation, I'd say it's kind of a blurred line. Speciation events happen naturally as well throughout history. Evolutionary history it's happened millions of times. We could potentially modify ourselves to the point that we can't reproduce with a human or a Homo sapiens of today's standards, but I guess you have to weigh the pros and cons. Would you rather live with a crippling, debilitating disease or use the technology. All right, no, I shouldn't propose it like that. It's not a dichotomy. We're going to be changing and evolving regardless through natural mutations and genetic drift and all sorts of things. So this is just a tool to help us not stumble so much along the way.
Well, so right now we're just talking about small changes in genomes as the present, so curing diseases and etc. And right now there are laws in place, so you cannot modify the germinal line. So these changes cannot be passed to the next generation. So that's what is in place. So right now, this is the scenario. But who knows, in the future, maybe we are willing to not design, but modify our genomes for better purposes. Maybe we want to colonize Mars, and we decide that we need several adaptations to live there because we are limited with our current bodies. Yes, another one.
That was actually along the line of my questions, combining genetics with space exploration. They're talking about the problems of going to Mars and radiation exposure and issues with bone loss in prolonged weightlessness. And has there been any thought given to the idea of using genomics to alleviate these things?
Well, maybe not directly, but maybe indirectly. So I know there were certain projects from the NASA to use hibernation in space travel, and they have quite a lot of research. So related to genomics, I can tell you that there is a little lemur in Madagascar that is capable of hibernation. That's the closest animal to us that is capable of hibernation. Actually, I've been studying it in collaboration with some people from the States.
He's being humble. He has 2 papers about this topic.
And this is the closest thing I can tell you about. But so far, I haven't heard anything. We're still in a very early stage. Nobody's thinking, okay, we're going to modify our genomes. We're going to change it to allow space travel. This is quite far. I think there are easier options, such as hibernation, for example, or other things.
Hello. Is there any work on the regulation of the bioprospecting? Because to modify an organism, you need to find something which has a trait that you want to use, unless your knowledge is really good in terms of writing it from scratch. And I know that there is synthetic life projects underway. So when it comes to the bioprospecting, one, with regards to the destruction of the biome which we're doing at the moment, and 2, what about the legal ramifications if you take a set of genes from a natural organism and you reverse engineer it? They've already established that you can't patent naturally occurring things. So vis-a-vis, how do you protect that, and also how do you regulate it from reverse engineering, where it could be applied harmfully?
Yeah, there are quite a few laws that already exist that prevent modifying, as Jose Luis mentioned before. CRISPR is still subject to all the regulations that already existed. We had these tools, they were just slower and more expensive before. So we're still subject to all these laws and regulations, and any proposed experiment has to go through ethical committees. But I think what you also asked was about being able to patent natural systems, being able to patent genes. And this is actually a subject of a lot of debate in the scientific community as well, because you could devote a decade of research onto a particular system to really, really understand it well. And you want to be able to patent a particular use of that system while not preventing other people from being able to use or study that particular sequence. And it's tricky, right? You don't want to be able to patent natural phenomena, but you also want to, some people want to be able to make money off of their research that they've slaved over for. So it's hard to pin it down. I don't know if I answered your question or not, but. All right, well, thank you.